Hi, I'm Dallas Kreisa, a graduate research assistant in the Institute of Plant Breeding, Genetics, and Genomics at the University of Georgia, and in this video I will demonstrate how to use a soybean seed drill. The purpose of the seed drill is to collect a small amount of tissue from the seeds by drilling a hole through the middle without destroying them. The tissue can be used for DNA extraction and running genetic markers to make trait selections before planting all of the seeds. The seeds will still germinate, just with some holes in the cotyledons. The seed drill can replace the seed chipping method shown here that was previously used for tissue collection, because it is safer and less tiring for the researcher's fingers. Before beginning, make sure you have all the materials you need, including 1.4 milliliter matrix tubes for the tissue collection, a latch rack plate for the tubes and a cover, a 96 well seed plate to put the drilled seeds into and a cover, 2 printed copies of a plate layout for both the latch rack and the seed plates, the seed you are collecting tissue from, a small brush, tweezers to remove the seed from the plate, and of course, the seed drill itself. Shown here is a sample tray that has 12 spaces to load the seed at a time, although today I am only going to show a single sample. Also, before beginning, make sure that the air compressor is hooked up and clean off the drill for any seed residue with the air compressor or a small brush. To begin, remove the sample tray cover. Lift up the seed cups to add the 1.4 milliliter tubes. After loading all of the tubes, place one seed in each of the seed cups for drilling. The seed should be laying flat on its side and centered. Return the sample tray cover. Rotate the turntable until the first sample is under the drill by pulling the lever on the side. To start the drill, hit the green Start button on the top. To avoid cracking the seed, lower the drill with the lever on the side and slowly push it through the seed. After the initial pass through the seed, raise and lower the drill bit a few times to ensure enough seed tissue falls into the tube. As you raise the drill bit up, use the foot pedal to blow air onto the drill bit to remove residue seed tissue. Turn the drill off using the red Stop button. If there is any seed tissue remaining on the drill bit, use a small brush to get behind the safety guard and clean it off. This is when you can rotate the turntable to drill the next seed, and repeat for all of your loaded samples. After you finish drilling all of the samples, remove the sample tray cover and take the first seed out of the seed cup. Looking at the seeds, the drill hole should be in the middle, so it should not harm the embryo of the seed that will develop into the plant. Take the seed from the seed cup and place it in the appropriate cell of the seed plate. The tweezers can be used to remove the seed if it is misplaced. Then take the matrix tube and place it in the matching position on the latch rack tube. It is super important to make sure the seed and the tissue are in the corresponding cells of the plates, so once the tissue is selected based on the genes that are present, the correct seed can be retrieved and planted. After the seed tissue is used for DNA extraction and to run genetic markers, the breeder can look at which genes are present in each individual seed and grow only the seeds selected by going back to the storage plate and selecting specific seed. The selected seed will be planted and grown as normal, but with small holes in the cotyledons from the drill. Being able to select seed based on traits present allows breeders to have smaller population sizes or breed for more traits and use their resources more efficiently.